Hi, and welcome back to my channel. In today's video, we are going to be discussing the cell cycle as well as mitosis. These are topics that are found in Biology 1, however, it is often reviewed in other biology courses. If you enjoy the content of this video, I ask that you please subscribe to my channel and like and share the video so that I can continue to make content for you. If you have any questions, please make sure to drop them in the comments and I'll get back to you. In a cell, there is DNA. DNA in a cell constitutes the cell's genome. DNA molecules in a cell are packaged into chromosomes. DNA includes all the instructions for the cell, and if the cell were to replicate its DNA, it would then be able to replicate its cells. Somatic cells, or body cells, have two sets of chromosomes. Humans have 46 chromosomes, resulting in 23 pairs. Gametes, which are reproductive cells, such as sperm and eggs, have half as many chromosomes as somatic cells. In humans, that would be 23. As I said before, in order for a cell to divide, the DNA is replicated and the chromosomes condense, as you see in this picture. Each duplicated chromosome has two sister chromatids. So what you see here is in each chromosome that you see, each portion of this would be a sister chromatid, and each one of these would contain the same information. So a sister would be a duplicate of the other one. So this would contain, one sister would contain all the DNA, and the other sister would contain all the DNA. So individually, these would be sister chromatids, and the entire thing would be referred to as a chromosome. The narrow waist that you see here is called the centromere, and this is where they are attached for the time being. In a eukaryotic cell, cell division is going to consist of mitosis, which is the division of the nucleus, including the genetic material that is in there, and then cytokinesis, which is going to be the division of the cytoplasm. We are going to talk about both later on in this video. Before we talk about the dividing of a cell, let's talk about the cell cycle as a whole. Now, the eukaryotic cell cycle has 5 main phases that I'll discuss in a minute, but I want you to understand that not all cells are going through this cell cycle at all times. If you look real quickly at this figure, what you'll notice is that there are several phases, which we'll talk about, and included in there is where the cell will actually divide. Now, there are some cells, like your skin cells, that are constantly dividing and undergoing mitosis so that you can create new skin cells, but there are other cells in the body, such as neurons, that are not dividing. There's another phase not shown here, referred to as GO, and that phase is the resting phase. It's a quiescent, non-dividing stage, and actually, most human cells are actually in this phase. Many human cells may never leave this GO resting phase, such as neurons, because they're not going to divide. However, some cells can leave it in the case of injury, where other cells need to be replaced, and they can then enter into the cell cycle we're going to be talking about now. So the 5 main phases of the eukaryotic cell cycle are the G1 phase, the S phase, the G2 phase, and then mitosis and cytokinesis, which would be the mitotic phase together, and this would be the area where the cell would divide. Now, mitosis is a separate entity, and then cytokinesis is a separate entity, and we'll be talking about how they both happen. Together, both mitosis and cytokinesis would result in a cell dividing from 1 into 2, so that they would be identical copies to one another. So let's take a look at each part of the cell cycle, okay? So the first phase is referred to as the G1 phase, or the gap phase. This phase is really important because it's the primary growth phase of the cell. The term gap phase refers to it's filling the gap between cytokinesis and DNA synthesis, which is going to happen in the next phase. For most cells, G1 is actually the longest phase. The next phase that the cell will go into is the S phase, or the synthesis phase. In this case, we are referring to DNA synthesis. This is the phase in which DNA will be replicated. Those chromosomes that we referred to before are going to replicate into two sister chromatids so that each chromosome will have a copy of itself. They are going to be attached together at the centromere I just referred to on the other slide. If you need a refresher on how DNA replication occurs, please refer to my other video on DNA and DNA replication. We are not going to go into detail on this here as I referred to it before, but DNA is going to be replicated in all those processes that occur during DNA replication, and that's going to happen in this S phase. The next phase that the cell will go into is the G2 phase, or the gap phase 2. This is where the chromosomes that were just synthesized, those sister chromatids that were just synthesized in the S phase, they are going to condense in this second growth phase. This phase is going to fill the gap between DNA synthesis and mitosis. During this phase, all the necessary components for cell division to occur are going to be made, such as mitochondria, other organelles. They're going to all replicate so that when the cell divides, each cell is going to have ample material and organelles. These three phases, G1, S, and G2, together all consist of what is called interphase. This is the phase of growth for cells and DNA replication. The fourth phase of the cell cycle is mitosis. This is division of the nucleus, where the chromosomes are going to separate. Those chromosomes that just replicated themselves, they are going to separate. And then finally, followed by the fifth cycle, or the fifth phase of the cell cycle, which would be cytokinesis, and this is where the cytoplasm is going to split, resulting in the formation of two daughter cells that are going to be formed from that initial parent cell. We are going to talk about these two phases in detail in the next slides. So we finally get to talk about mitosis, which again is the division of the nucleus, including all the genetic material that is in there. In this video, we are going to divide mitosis into 5 phases. Some simplify it and divide mitosis into only 4 phases. Some books will elongate that out and add or early and late phases of each one of the phases. But for this one, we're going to divide mitosis into 5 different phases. They are going to include prophase, prometaphase, metaphase, Anaphase, and Telophase. Like I said before, some will just simplify that into 4 different phases, being PMAT, and will not include this prometaphase that we're going to discuss. But we're going to discuss the 5 different phases and what occurs in each phase. So let's start with prophase. Prophase is the first phase. Remember, at this point, the chromosomes have duplicated themselves. The cell has moved from interphase now into the mitotic phase. And so in prophase, the chromosomes are going to continue to condense. The centrioles are going to move to each pole of the cell. And the centrioles are a really important cylindrical organelle that is in the cell. And in the G2 phase of interphase, they have replicated themselves. So there's actually 2 pairs, and they're going to move to opposite poles of the cell. So the centrioles are going to move towards opposite ends, and here they're kind of be moving away from each other to eventually get to the opposite ends. And they lie 90 degrees to one another. And then from them, the spindle apparatus is going to start to be assembled. So it's going to kind of look like this, and they're going to continue to move. The other thing that's going to happen during this phase is that the nuclear envelope, which surrounds the genetic material, is going to start disappearing because it's going to be dissolving and being absorbed by the endoplasmic reticulum. This will move the cell into prometaphase. And in prometaphase, the chromosomes are going to become attached to the spindle apparatus by their kinetochores. And the kinetochores are part of the centromere. The centromere is going to be that inner portion where it kind of thins at the waist, and they're connected, and they are going to start connecting to that spindle apparatus. So as these chromosomes attach to the spindle apparatus, they're going to be oriented in such a way that the kinetochores of sister chromatids are attached to microtubules from opposite poles. And so you can see how I've drawn them here. Now, first of all, understand that there aren't 4 pairs of chromosomes, okay? Remember, we talked about that there's 46 chromosomes in a human cell. That means there's 23 pairs, but there would be 46 of these that would have duplicated themselves in order for the genetic material from the cell to separate into each. So I'm not going to draw 46 of these because that's a lot. So we're just going to show 4 to kind of represent this. But imagine in a human, there's 46 chromosomes in a somatic cell, and so each one of those has to replicate and then split so that the cell, when it divides into 2 daughter cells, they each have all the genetic information that they need. But the way this is set up is that the spindle apparatus comes down from the centrioles, and they are going to attach each to one sister chromatid, so that when they're pulled in opposite directions, they're taking 1/2 of that chromosome and the genetic information. So you'll see that they're oriented, so they're going to take 1 from each on one side and then 1/2 from the other side. That way, when they split, the genetic information will split equally. At this point, the microtubules from the spindle apparatus are going to begin to pull each chromosome towards the center of the cell, and this is going to eventually end up in our next phase. If there are mistakes in this microtubule placement, that can be fatal. So for example, if all the microtubules attach to the same pole instead of opposite poles, then when the information divided, when the chromosomes split, there would be unequal information in each cell. So it's very important that they arrange in this way where one sister chromatid is going to go to one part of the cell, and then the other sister chromatid will be directed towards the opposite pole. At the point of metaphase, the chromosomes line up at the equator where each sister chromatid is oriented to opposite poles. The centromeres align on an imaginary line called the metaphase plate. This is one of the easiest phases of mitosis to identify, as all the chromosomes are lined up in the middle. As you can see, each sister chromatid is facing an opposite pole. The cell is now ready to begin the separation of these sister chromatids. In anaphase, the proteins that are causing the centromeres to stay together are removed. These cohesion proteins are removed, and this allows the centromeres to separate. In that way, the microtubules can pull the sister chromatids towards the opposite poles. So in anaphase, since the sister chromatids are now separated, what you can see here is that the microtubules are going to start pulling these sister chromatids. They've now separated one from another. Remember, each one contains all the DNA from that chromosome. So they're each an identical copy to one another, and each half is getting split and now being pulled to the opposite sides of the cells. Telophase, the last phase, is almost the complete opposite of prophase, where everything started. In telophase, the spindle apparatus is going to disassemble because now the chromosome information is on separate sides of the cell and it's not needed anymore. The nuclear envelope is going to form around each set of sister chromatids, and the chromosomes are going to begin to uncoil. The nucleolus is also going to reappear in each new nucleus. As you look at this picture that I've drawn, you'll notice that it looks as if there are two cells in one, which there basically are. So at this point, at the end of mitosis, the nucleus has divided, each having its own set of DNA now, and eventually the nuclear envelope that you see I've drawn here reforming will completely form. So the last thing that needs to happen is that final phase of the cell cycle, which is cytokinesis and the splitting of the cells to make two. So right now we have two nuclei in here. There's one nucleus and another nucleus, and now we have to split the cell so that we have two identical. So in cytokinesis, now that we've finished mitosis, in cytokinesis we have all the genetic information in two different nuclei that are here. I've shown the chromosomes uncoiled in here. The nuclear membrane is now reformed. Obviously, each cell has all its organelles, etc., that is necessary. I'm not going to draw them all, but what happens is this cleavage furrow is formed, and you saw that in telophase as well. But this cleavage furrow forms, and the cell is actually going to be cleaved at the cleavage furrow, resulting in the formation of two separate cells. Once this is completed, we now have two identical daughter cells. So these cells that are formed are identical to one another. So if it was a skin cell, they would be identical to one another. And this process would conclude the division of the cell. So mitosis, the division of the nucleus, and cytokinesis, the division of the cytoplasm, is going to result in the formation of two identical daughter cells from that initial parent cell that we started with. If you couldn't follow my crazy drawings, or you just wanted to see it in a more artistic depiction, this shows the different phases that we just went through. You can pause this here to look at them in more detail, but quickly, this picture over here starts with the G2 phase of interphase. So this is the second growth phase, still in part of interphase, and then moving into mitosis after that. So then you would see the prophase, prometaphase, metaphase, anaphase, and then telophase, which would be followed by cytokinesis. Remember, telophase is the last phase of mitosis. Cytokinesis is its own phase where you would have the splitting of the cytoplasm. These images show cytokinesis. On the left-hand side, you can see the cleavage furrow forming, and this results in the cleavage of the cytoplasm, and so there would be two daughter cells formed after the cytoplasm is completely disconnected between the two. This happens in animal cells where we get a cleavage furrow. On the other side, you see how plant cells divide, and in plant cells, rather than a cleavage furrow, there's a cell plate that actually forms between the two cells. And so you can see here the formation of the cell plate, and then there's a new cell wall, and then the daughter cells. This is different because in animal cells, the cells are separate from one another, whereas in plant cells, you can see that they are still adjacent to one another. Now, because there's a lot of things going on in the cell cycle and things being replicated like DNA, organelles, etc., proteins being made in order for all of this to happen, there has to be some control of the cell cycle. And so briefly, we're going to talk about how the cell cycle is controlled, mainly by three different checkpoints that we see throughout the cell cycle. So the first checkpoint that I'm going to refer to is the G1S checkpoint, okay? And this is going to be the first checkpoint that we see here. And this is going to be the checkpoint that the cell decides to divide. If the cell receives a go-ahead signal at this checkpoint right here, right before it goes into S, it will usually complete the S, G2, M phases and divide. It will continue to go through. However, if it does not receive the go-ahead signal at this checkpoint, then it will exit the cell cycle and switch into a non-dividing state called the GO phase. So this checkpoint is really important, this G1 checkpoint, because if it passes it, then it can continue through. If not, then it can go back over to the GO phase. The next checkpoint that we come to is the G2 checkpoint. This G2 M checkpoint, right between G2 and M, is going to be where the cell makes the commitment to mitosis. So if something went wrong with DNA replication in the S phase, etc., the cell can still stop. Once it passes this checkpoint, then it's going to make a commitment in order to go through mitosis. So this G2 checkpoint, once passed, mitosis will occur. If it doesn't pass, then the cell won't go through mitosis. And then finally, within the mitosis phase itself is one last checkpoint, the M checkpoint. And this is the metaphase checkpoint. And this is the really important one because remember we said before that there's a lot of issues that can occur during metaphase if the spindle fibers aren't all attached to a sister chromatid to pull them to separate parts of the cell. So this is going to be the checkpoint to make sure that all the chromosomes are attached to the spindle fibers and that they're oriented in the correct direction. And then once that happens, we can continue with mitosis and then eventually cytokinesis. There are certain enzymes and proteins that are needed at each checkpoint in order to proceed in the following phase, and these are all within the cell. We're not going to go into detail on those, but they're important in each phase at each checkpoint that these proteins and enzymes are present to allow the cell to go through all of these checkpoints. So this just ensures that the cell cycle is controlled and that the cell can divide in the way that it needs to divide. If there's a loss of these cell cycle controls, such as what we see in cancer cells, then they can divide uncontrollably with no checkpoints, okay? So for instance, cancer cells do not respond normally to the body's control mechanisms. They may not need growth factors to grow and divide, and so therefore they are growing uncontrollably, basically one on top of another to form these tumors. I want to thank you again for watching my video and remind you that if you have any questions, go ahead and drop them in the comments and I will answer them. Thank you.